All right, and good afternoon, everybody. Welcome to the Wolfpack Wellness Researcher Web Series. I'm your host, Tremaine Lee, standing in for Dr. Susie Gazelle, and I am the Assistant Director of Wellness for Wellness and Recreation. So nice to see everybody. Nice to see some returning faces. But this series is a collaboration between the university's Wellness Advisory Committee, the NC State Libraries, and NC State Wellness and Recreation. And we aim to discuss wellness research that is happening across NC State's campus. So for those of you joining us live via Zoom and or YouTube, you can submit questions to our speaker using the chat function, and we will have Q&A at the end of our show. So as questions arise, be sure to just kind of drop it in the chat, and no need to be timid. And so today's guest is Dr. Rodolphe Berengu. So welcome, Dr. Berengu.
Good afternoon, everyone. Thank you for having me.
Absolutely. And we are just excited to hear from you today and learn more about you. And so I'll just read a little bit of your bio. Dr. Berengu is the T.R. Clanhammer Distinguished Professor in Probiotics Research at NC State University. His research focuses on the characterization of CRISPR-Cas systems and their applications in bacteria and beyond. Dr. Berengu spent 9 years in research and design and MMA at DuPont. For his CRISPR work, Dr. Berengu received several international awards and has been elected into the National Academy of Sciences, the National Academy of Engineering, and the National Academy of Inventors. Additionally, he has been affiliated with multiple startup companies and is the co-founder of Intellia Therapeutics, Locus Biosciences, Trico, CRISPR Biotechnologies, and Ancilia Biosciences. So welcome to our show, Dr. Berengu, and thank you for joining us today.
I'm glad to be here, and I'm excited to share some insights and give some thought-provoking takes to the audience.
Absolutely. And so with that being said, can you tell me a little bit about you and your research lab?
So I'm actually twice an alum of NC State. I did a master's here, I did a PhD here. I went to grad school a number of times. I don't need to go into too many details, but I'm back home where I went to school. And as you mentioned, I have the great distinction to be the Todd R. Clanhammer Distinguished Professor in Probiotics Research. And as a matter of fact, I did my PhD in the Clanhammer lab. So I know NC State quite well. And I went to industry for 10 years, and I came back about 9 years ago. And I run the CRISPR lab. And what we do in the CRISPR lab is use, deploy, characterize, harness, and apply all kinds of CRISPR-based technologies. And for those of you who haven't read Walter Isaacson's latest book, The Code Breaker, for those of you who haven't seen as of yet Human Nature, a documentary on CRISPR and gene editing on Netflix, or for those of you who haven't heard as of yet about the revolution that is occurring before us in real time with regards to CRISPR-fueled genome editing, this is what we work on. So it's a very disruptive, very democratized, very powerful technology that enables virtually the average geneticist. So there's hundreds of people at NC State who use that on campus to manipulate almost trivially, but not quite, the genomes of any species you can think of across the tree of life. So you mentioned some of the companies I've worked with. We work with bacteria, so very, very small. We actually even work with smaller organisms like viruses. So we can use CRISPR-based technology to rewrite the genome of small things like viruses and bacteria. And then we can deploy that in organisms of interest to us. Think humans, think livestock, think crops, or we can go all the way to very large genome and very large organisms like trees. And again, my lab and many other labs on campus are using those very powerful, convenient, and affordable genetic surgery tools to change genomes. And the way we do that, if I have a minute, the way we do that is to use CRISPR systems that in nature are DNA-encoded molecular machines that provide immunity. So in humans, we have antibodies and antigenes, lock and key, right? Antibodies. And through the vaccination process, we have protein-protein interaction that enables us to distinguish cell from non-cell and build three-dimensional protein-based immunity against infectious disease and viruses and foreign elements. Bacteria are not as sophisticated as we are. So they nonetheless have an immune system that's called Crispr. So Crispr in nature is an immune system that enables them to do that. But instead of being protein dependent, it's DNA dependent. And they have a CRISPR molecular device that acts kind of like a phone that will take a genetic picture of an invasive genetic element, like a virus, like a transposon, like a plasmid. It will take a genetic picture of it. It's like a short word of 30 letters, and it will integrate it into its CRISPR for sequence-specific targeting and cleavage. That's what CRISPR is in nature, but nobody really cares. What's really cool about CRISPR is what you can do with it. And what people are doing with CRISPR is using that molecular machine to have targeted, programmable, sequence-specific DNA cleavage. Think of it like a molecular scalpel, like a razor. And most of the people in the audience have used a razor at some point in time to shave a part of their body we don't need to hear about here. And what happens when you shave is occasionally you cut yourself. So that's what CRISPR does. It actually cuts DNA, but it's very precise. Like a molecular scalpel, you can do Control F, have a series of 30 DNA letters that you look for, and it will precisely cut 3 letters away from one end of the word. And when you cut that DNA, much like when you cut ourselves, DNA repair occurs, like skin repair occurs for us, and you get a little scar. So genetically, CRISPR enables geneticists to change DNA, to scar DNA, to alter DNA, to edit DNA precisely and programmably at a location we can determine. So think of the human genome, 3 billion letters is the equivalent in many ways of Sir William Shakespeare's masterwork and complete works. You can do Control F with CRISPR. To be or not to be, that is the question. You find that word in the book of life, 3 billion letters. And then we cut between that and is, and we can change the text exactly right there. To be or not to be, that's the question. To be or not to be, hashtag the question. To be or not to be, the quintessential question that defines humankind. You can insert DNA, you can remove DNA, or you can rewrite DNA. And what's really cool about CRISPR, as you can imagine, is that we have the ability to use that technology to rewrite any kind of DNA you can think of in any organism you can think of. Again, I mentioned viruses, I mentioned bacteria, I can mention livestock for breeding, crops for breeding, obviously humans for genetic disease, all the way to CRISPR trees we have on campus in the greenhouse. And we can engineer and or alter DNA and or correct DNA to cure disease or breed for enhanced traits, enhanced features, enhanced benefits, and on and on and on and on. And what's great is that it's very convenient, it's very powerful, it's very doable, it's very affordable, it's very precise, it's very specific, it's very efficient. So the issue is how people use it. And people with the right motivation and the right mission and the right mindset can cure genetic disease, can breed a more efficient crop, can breed a leaner pig or a pest-resistant cow or an infectious disease-resistant turkey, right? Or a more sustainable tree. Or there's some questionable scientists out there who are using this to think of designer babies, or to bring back the woolly mammoth, or to, engineer viruses to be more pathogenic. And that's the conundrum of a great technology like CRISPR. There's a lot of excitement to be had therapeutically in food and ag and biotech and anything that's related to human health, whether it's your diet or your microbiome or your genetics or your physical performance or your mental health. But there's also a lot of consequences about the ethics and the regulation and the implications and PR and scientific skepticism and the discomfort that a lot of non-experts have about, quote unquote, the ability of scientists to play God and engineer humans. or the dystopian and questionable, ethical or unethical applications that may be out there. So there's a lot of aspects to that. And what we do on campus in my lab is we develop novel technologies. We develop better tools, more, like sharper scalpels, more precise and nimble molecular scissors. There's different versions of CRISPR where we can have like a chainsaw that really like removes, so you don't want to shave with a chainsaw, right? Like you can really take things away, or we can be very, very, very precise, or we can be nimble like a Swiss Army knife. And so we work on those technologies. And then of course, we deploy those technologies in bacteria. So I work in the food science department, the same as Professor Goodell, and we engineer microbiomes. We make gut microbiomes healthier. We make vaginal microbiomes healthier. We make food microbiomes more efficient, more consistent for yogurt flavor or cheese texture, or think of things like fermented like kombucha or pickles or sauerkraut, or of course, fermented vegetables. So, and fermented alcoholic beverages. There's a lot of things we can do with bacteria and microorganisms that are great and that we do on campus. But also we work with a lot of people that deploy those technologies in the clinic, or deploy those technologies in the field for agriculture, or deploy those technologies in trees for forestry. We have a great forestry department, for example, at NC State. So we work with probably dozens of labs across the world to deploy and harness the technology to breed more sustainable processes. And then, of course, we also work with industry. So we have strategic partners that use and deploy those CRISPR-based modalities to treat disease in patients. We don't have a medical school at NC State, so we're going to work with other people to bring that to the field and scale it up, to bring that to the food market and scale it up. You mentioned DuPont. I worked at DuPont for 10 years. And that's why we do some entrepreneurship, and that's why we do some patenting, and that's why we do some PR, and that's why we don't just quote unquote do the science. I mean, science is hard enough for sure. But what I like about that topic, and presumably one of the reasons I'm here to talk to the audience, is there's so many applications that we can talk science all day. And also there's so many implications that we can talk philosophy and context and science communication and risk management and societal implications. So I'm quite excited to be here, and I'm sure there's a lot of things going on in your mind, and I can't wait to get a Q&A.
Yeah, absolutely. And I know you covered a multitude of even some of the questions that we were going to ask today. So that is quite impressive. Loved your opening. I really appreciate it. One of the things I kind of want to dive into is when we talk about our elements of well-being, we always talk about purpose and how purpose really drives your passion. And so what about working with CRISPR in your studies, and what kind of spurred your study, and how does it tie to your passion and your purpose?
Yeah, so I'm a food scientist, right? And let food be that medicine, and medicine be that food, right? So I like food. I eat food. I enjoy food. I don't want to splurge too much with food, and it's very enjoyable, but also very nutritious. And when I think about health, whether it's brain performance, athletic performance, overall personal performance, and engagement and sleep and lean body mass and whatnot, like nutrition is a huge part of that. So being able as a microbiologist to use CRISPR-based technologies and deploy CRISPR technologies and apply CRISPR technologies to enhance all kinds of food aspects, whether it's sustainability or efficiency or flavor or texture or food safety and microbiology, or the human microbiome and gut health and even vaginal health and skin health and oral health. It's pretty exciting to be able to touch all kinds of different aspects like food with one common denominator that we're passionate about, that you can smell and taste and put in your mouth and play with. And I think there's like a visceral, humans like food kind of immediacy that's obvious. And I'm passionate about it from a consumer standpoint because I love food and all kinds of food. And also I'm passionate about it from a scientific standpoint, because there's a lot of aspects to that. It's not really one dimensional, right? Now, more philosophically, what's great about food is that it has implications. Like, how much do you enjoy food? Too much. Do we all have access to food? And there's an equity aspect to it. There's a socioeconomic aspect to it. There's a cultural aspect to it. Can you consume alcohol and not consume alcohol? And if you do you like wine? Do you like beer? Or do you like champagne? So there's a lot of like interesting societal, cultural, science-related, but not science-centric aspects that we get to talk about all day, every day, in the context of food, in the context of science, in the context of cooking, in the context of health, in the context of industry, in the context of consumers, in the context of MDs, in the context of pets and livestock and agriculture. And that, to me, keeps it fresh, pun intended, right? It keeps it exciting. And, I can change my career a little bit, or I can have, 10 Zoom meetings back to back to back to back to back all day, every day. But every single meeting is going to be a little bit different. It's not so redundant. It's not always the same. I can have a philosophical discussion 1 hour. I can have a financial discussion the next hour. I can have a genetics discussion, deep dive for 2 hours. And I can go home and talk about it when I eat the food. And that, to me, keeps it kind of energizing and fresh and all-encompassing in ways that actually very few topics enable us to do that. And that's why I love being a scientist. As a matter of fact, it's kind of funny because there's no such thing as food science. There is no science of food. It's many sciences applied to food, right? Like, it's like anything you can think of. It can be engineering, right? It can be microbiology, or it can be sensory, or it can be chemistry, or it can be physics, or it can be even marketing, or it can be manufacturing, and it could be financial, and all those things, but apply to food. And that food, visceral, basic, elemental passion that we all have as consumers, right? Because if you don't eat, you're not going to make it. And if you don't eat, you get hungry real quick. And that is like an emotion that we can't control. It's animalistic. It's in our DNA. It's in our cultures and gathering and, foraging and, agriculture and culture. I mean, it's amazing to me how that one aspect touches everything that we do and makes it kind of all encompassing in ways that are very excitable and that I'm excited about.
Awesome. Thank you for sharing that. I mean, there's so many magnitudes, and it's just a very dynamic topic in general. So yeah, that's amazing that being just, a food scientist and the depth of, what you want to study, and additionally, like the benefits that you can discover by doing your research is just absolutely out of this world, to be honest. And, I think well beyond this time, but definitely moving in the direction that's going to really benefit our future generations when it comes to health and well-being. So super fantastic. One of my next questions is, what is the coolest and most interesting thing about this research that you found?
Oh, it's an interesting question. So what's amazing to me is how, bacteria that are so small, they're invisible to us, right? They're microscopic. That's why they're microorganisms. We can't see them. We can't decipher them and understand them unless we use fancy techniques. What's amazing to me is how complex and sophisticated the invisible can be. How like mesmerizing it is that you can't see it, but you can maybe smell it or taste it or feel it or analyze it or investigate it. And if you really zoom in all the way into actual DNA, not just the organism, but its very core quintessential piece, DNA, I am so mesmerized by the ability of DNA to be so nimble, so functional, so potent, so predictive, so manipulatable, so engineerable. Like tinkering with DNA is so cool. And what the ease with which we can change the living is unbelievable to me. Like, I had no idea that we could, write synthetically new organisms that don't exist.
Wow.
That the average geneticist can do it almost in a heartbeat, and then go in and rewrite how organisms make yogurt or cheese, or how they make us sick, or how they make us healthy. Like the speed and ease with which we can change their DNA is hard to just comprehend, right? Like you rewrite and change the Word document, we can do that in the DNA of organisms that live and kind of control them to some extent. So I like that. Like that's amazing to me. I can't believe it. What's really unbelievable, though, to answer your question, is that we can do the same in humans. Like that just blows my mind. And 6 years ago, right, I founded a company called Untitled Therapeutics. I co-founded a company called Untitled Therapeutics, and it came out of that dream, which I thought was a crazy idea at the time. We can use CRISPR in humans to cure genetic disease, and it was kind of like a dream, right? Like a hope that we had. Maybe one day we can use CRISPR to correct genetic disease in a human, but it's going to be hard and it's going to be slow, and we have a high chance of failure, and it's going to be difficult, and we're going to need a lot of things to really go right to get there. And what's unbelievable is that we managed to do it, and we already have clinical trial data, and we've been able to show in patients that live, right? Like in FDA condoned IND enabled studies.
Wow.
That we can go into the liver of a patient and deliver a CRISPR and change enough and correct enough of that DNA so that a disease will go away. And it's supra therapeutic, and we restore their health. Like that just blows my mind, to answer your question, that we can actually not think of doing it, which is already crazy in and of itself, right? To have the idea, it sounds like a science fiction movie, like we're going to go into people's DNA and we're going to correct their DNA and change their traits. Like you see that on TV, like they're going to change the eye color, or the height, or the muscle. They have all this like the crazy stuff. It makes no sense, right? But that we actually go into a patient liver and change enough of their liver cells that their liver disease goes away, that just blows my mind. It shouldn't, right? Because that's what we do it in the first place. But the fact that we were able to do it so efficiently and so fast is just, wow, I can't believe this.
That's super impressive. Yeah, that's really impressive. That's kind of mind-blowing to me. Like I was kind of stuck at the when you were talking about manipulating organisms, bacteria, kind of takes me back to the mindset of like making medications. When you trick bacteria to make insulin, for example, I'm assuming that's the same type of technology that's being employed there. So we actually have a question from the audience too. It looks like Cindy Levine. And so Cindy's question is, so is your lab working primarily on developing the tools, and you collaborate with other departments for more specific research projects?
So yes and no. So when it comes to the microbiome, we do the tools and we do the applications in my lab. Like we can make bacteria more amenable to being immunogenic, or less inflammatory, or to reduce inflammation, or to trigger a vaccination event, or to ferment milk faster, or to make cheese taste better and stuff like that. So we can do that in bacteria. Again, it's not trivial, but if you get a PhD in my lab, 6 years later, I can turn it into a CRISPR wizard and change the genetic content of bacteria to enhance food, pet, or human health. Now, if we want to do that in humans, you're not going to see that in the CRISPR lab at NC State. We have to work with people who have access to patients and access to hospitals and access to clinics and companies that can deploy and meet and manufacture medicines, experimental medicines that will be injected into a patient. That we have to work with others. Or we can work with other labs at NC State, like Jack Wang's lab, to deploy CRISPR and grow trees, right? In which we change lignin content so they're more amenable to sustainable paper industry and pulping.
Awesome. Thank you for that. And we have another question by Alexander Hall. What would be the main jobs that would be working with CRISPR, and how would they use CRISPR in their respective fields?
Yeah, so it's R&D jobs in the industry, right? Research and development jobs in the industry. The last 14 people who graduated from my lab with a PhD or a master's all went to industry. Most of them went to the pharma industry. Some of them went to the biotech industry. A handful of them went to the food industry. But it's really industry R&D job to develop products, develop therapies, develop technologies, breed organisms that are going to go somewhere in the pharmaceutical industry, the food industry, or a farm.
Awesome. So my next question looks like it's coming from David Hunt. Could CRISPR be used to reduce virulence of a virus like flu, or does the flu have too many strains to be a viable target?
Yeah, so it's kind of interesting because in nature, like I said at the outset, in nature, CRISPR is an antiviral. Like that's what it actually does originally and functionally. So CRISPR is one of the best, most nimble, efficient antivirals on planet Earth. Now, some of the and from a technical standpoint, CRISPR is very good at targeting double-stranded DNA viruses. So things like the flu are not double-stranded DNA viruses, they're RNA viruses. So COVID is an RNA virus. So we can use other CRISPR tools than the popular Cas9, for example, to indeed be antiviral. And they're very programmable, but how efficient they are, how deployable they are, and how spreadable they are, and how much or not a virus will respond and adapt to an antiviral response is what we're working on and other people are working on. So there's a whole interplay and gamesmanship that happens, action, reaction, ecological consequences. We see that with COVID, we see that with the flu. That's why the flu changes every year, right? It mutates so fast. So absolutely, we can use CRISPR to target conserved sequence of viruses that drive them by mutation to extinction, but it's not trivial. And there's a lot of people working on, I mean, there's about 200,000 labs in the world working on CRISPR. Wow. I'm assuming probably 2 to 3% of those people, which is a large number of people, are working on CRISPR antivirals. There's a lot of promise. The question is not whether it can work. The question is how well it can work in the whole human. So we can cure, in one cell, we can cure in a test tube, but if we have viruses all over our whole bodies, the dosage and the delivery is a challenge.
Awesome. Well, unfortunately, I know we have some more questions in the chat. And so what I would do is I will save this chat. I know AG had a question, Zachary had a question, and I'll send it to you, Dr. Berengu, and definitely get your response. I definitely want you to get your questions answered. But that is all that we have time for today. And so again, I want to thank you, Dr. Berengu, for your time today and just speaking with us about your research on CRISPR. Is absolutely fantastic, super enlightening. And I really can't wait to see what all CRISPR does in the future and as it continues to evolve and just be a more incredible tool over time. So I want to thank you all for watching. And I want to remind you that we broadcast this every third Friday of the month at 2 p.m. in Eastern Standard Time. And so next month's show will include an interview with Dr. April Fogelman from the Department of Food, Bioprocessing, Nutrition Sciences. And she'll be talking about her work related to breastfeeding support. So you can find more about our Wolfpack Wellness Researcher web series and previous recordings at Wolfpack Wellness. Really, it's wellness.ncsu.edu. So that is the website. And so I am your guest host, Tremaine Lee. Go Pack. And until the next time, be well, everybody. Take care.